Welcome, everybody. Thank you for attending our workshop for the spring this year. We have a lot of exciting talks that we're going to showcase today. And I'm just going to give you a little bit of a recap on what we've been up to and what the goal and the methodology for today's talk are going to be. Last fall, we had an exciting release of our latest version 7.0, and we went over the differences and the journey that it took to get from our older version, version 6.x to 7.0. We showcased the new look, the new features, and the new improvements. For today, we're going to talk about what's new. We have some existing feature enhancements that we'd like to show you, as well as some new capabilities from our existing features. But also, we're going to look forward towards the future. What is next? And Jason and Senna will talk about AI integration within our products and what we're looking forward to working on moving forward. So welcome and thank you for attending. So I'll be giving the first talk for today. It's called Conjugate Builder for ADC, which we've acclimated to any drug conjugate instead of antibody drug conjugate. And you'll see what I mean as we go through the slides. So one of the things that has been coming along in recent years is the next generation of therapeutics. AstraZeneca has a really nice breakdown of the next generation on their website. I have the source below. And full disclosure, I took their screenshot from their website, but it really encapsulates the evolution of therapeutics that we've gone through in the drug discovery process. While small molecules still is an important factor in identifying new modalities for therapeutics, it has grown exponentially to include biologics. And this ranges from peptides, nucleotide-based therapeutics, antibodies, and cell-based therapeutics. With the explosion of this new generation of therapeutics, you have a little bit of a challenge in terms of informatics capabilities. So currently, many companies in R&D and drug discovery have legacy systems, either that they purchased early on or that they built themselves, homegrown systems. And it lands in either one or two specialties. The first is a chemistry-based legacy system, which can handle small molecules, because that's what the therapeutics were 10, 20 years ago. It's a relatively mature software system. A lot of companies have made very good products for it. But to this day, there are issues handling enhanced stereochemistry capabilities with these type of systems. As small molecules get more advanced, and as you strive to increase specificity over a kinase, for example, or minimize toxicity effects, you start to introduce often multiple stereocenters in a small molecule. And this presents a challenge for any software to try to handle this enhanced stereochemistry. On the other end, we have biologics-focused software. These are generally tend to be sequence-based, and for the most part, they only support natural amino acids and nucleotides. One of the challenges that we face is simply the integration between these two types of legacy systems. Oftentimes, what we see is that they do not integrate well, and companies either need to purchase one or the other or both systems for their respective departments in chemistry and biology. On top of that, it also neglects the rise of the next generation of therapeutics, right? The macromolecules that we were discussing earlier in the slide before, your peptides, your oligonucleotides, your lipid nanoparticles. This is missing in the legacy systems for the R&D. So how do we solve for that? How do you bridge this gap? On the left side, you see the small molecules, which are represented here in a SMILE string or in a mole file, which is machine readable. And for biologics, they're typically represented as a letter sequence. So the solution here is HELM technology. HELM language. HELM is an acronym that stands for Hierarchical Editing Language of Macromolecules. Now, my colleague Scotty in the next talk is going to go in depth on HELM, so I won't go too in depth on what this means, as they'll do a much better job than I can. I'll give you a quick preview on how we utilize HELM in our sketcher. HELM allows you to progressively build upon a small molecule represented by a single atom to a monomer, to a simple polymer, to a complex polymer. And you can see on the right-hand side, the machine-readable HELM language that is used to denote these types of more complex biologic structures. So Scotty's going to give a really nice talk after this one, going more in depth on HELM and the journey that we've had on molded HELM capabilities. But because of HELM, we're now able to create a conjugate builder. And this is going to allow us to address the next generation of therapeutics. I'm going to talk now about our conjugate building capabilities. Before I do so, though, what are conjugates? In this screen, I have examples of 3 different types of conjugate system. A conjugate is essentially a combination of 2 or more entities that are usually connected by a linker. On the left-hand side, I have an example of an antibody drug conjugate, which has a cytotoxic payload or a drug that is connected to an antibody via a linker. And the idea here is that the antibody will attack a target cancer cell with a specific antigen, and it can deliver the payload or the warhead to destroy that cancer cell. Similarly, we have small molecule drug conjugates. The most popular ones nowadays are PROTACs. These utilize the E3 ligase system that our body can use to then degrade proteins. And it's the same concept. You have a drug payload, a linker system, and an E3 ligase. Same can be said for bi and trispecific antibodies. These have the same goal to target a specific cell or a specific entity, but they're made up of different components with different entities. And it goes to show you how the next generation of therapeutics has increasing complexity. And that brings challenges with supporting conjugates. Legacy systems, aside from the challenges that I mentioned earlier, in which they're siloed either as just chemistry or biology based, they're also, many of them are unable to support the many components that are used to develop a conjugate. On top of not being able to support them, let's say they can support them. Now we have a challenge on how do you register with several different modalities, multiple modalities that is necessary to grab these conjugates and connect them together. How do you depict connectivity between these conjugate pieces? This is yet another challenge that we've faced in trying to register, for example, for ADCs. And finally, it's important to be able to track the relationship and the genealogy that each individual piece plays in the production or the construction of a conjugate. So with these challenges, Scilligence was able to develop our own conjugate builder. Conjugate builder starts by allowing us to register the components of said conjugates. In the screenshot here, I'm showing just a small piece of the modalities that our registration system, RegMol, is able to handle. So you can register a payload, a linker, perhaps a combined linker payload, or a warhead such as an antibody, a small molecule, etc., that is captured, registered, and has uniqueness check capabilities. Then you build the conjugate after you've registered these components. Our ADC builder is able to grab different components of your antibody drug conjugate that have already been pre-registered, and you can assemble them in the way that, in a unique manner, either with an antibody positioning, the conjugation method, or a DAR ratio. We then have a lineage or relationship metadata that's tracked after you've built the ADC. This tells you where each component came from, where it was registered before, and if it can be used repeatedly to build other types of conjugates, using same components and altering it by one different piece. And at the end, we get a result that looks something like this. In this example, we have an antibody drug conjugate in our sketcher, JSDraw. On the left-hand side, we have a registered antibody that actually contains the heavy and light chain sequences of that antibody, the connection to the linker, and then that connection to the payload. And this is all displayed in one sketcher that can handle both small molecule representation as well as biologics representation, in large part thanks to HELM. But what are the new features? We've worked on conjugation before. This is something that we've successfully had in our weaponry to discover new drug targets and new drug discoveries. But what's new? So our conjugation method is something that we're actively working on. This allows us to connect components based on a predefined business rule set. So you can say all these antibodies are connected to a cysteine or perhaps to a lysine by an amide coupling. In the past, the user would have to individually connect each piece to the either the linker or to the payload. So what this is allowing us to do, the conjugation method, it allows us to automate conjugate building. And it facilitates, for example, bulk registration. Imagine if you have 100 ADCs you'd like to register, the pain point of having to manually connect each antibody drug conjugate in the registration system for final registration and uniqueness check. With the conjugation method, you can upload batches of ADCs that have specific methods of conjugation. So it facilitates automation. Another new feature is our multi-payload ADC capability that we're working on. So over here, I have an example, a pictorial example of a multi-payload antibody drug conjugate. In this example, we have two different payloads with different DAR ratios. In this case, we have two MMAEs and then an MMAF payload attached to an antibody. What we're working on is being able to support dual or multi-payload antibody drug conjugates that also supports uniqueness checks, can identify the connection point, and display the DAR ratio of more than one payload. And down here, I have an example of what we're working on. I'm trying to support multi-payload ADCs. As you can see in this example, similar to the one I mentioned earlier, we have the antibody, we have our linker, only this time we have two of the same payload connected to the linker by a distinct connection points. So I hope we're working hard on trying to develop software and informatics capabilities that allows the scientist to move forward with this next generation of therapeutics. And this is just a small preview of what we wanted to show you for our conjugate builder. And with that, I thank you for listening to my talk. And I'm going to check if there were any questions that need to be read out loud or that were answered in the chat. All right, thank you.
Thank you very much, Alan, and want to extend a welcome for those who have just joined us during Alan's presentation. I'm very excited to introduce the next speaker. So the next presentation is by Scotty Blechman. Scotty is a manager on the Scilligence development team and has been with the company for just under 4 years. Prior to their career in scientific software development, they obtained degrees in biochemistry and chemistry and worked in both molecular biology and medicinal chemistry labs. So just to give just to introduce the topic, HELM is a widely used standard for representing complex biomolecules. However, converting molecules from MO file format to HELM notation is often a challenge due to varying monomer definitions, drawing conventions, and peptide oligonucleotide backbones. To address this, Scilligence has developed a commercial tool called multi-HELM that performs this conversion based on the specific monomer library defined by end users. The tool uses algorithms and fragment identification to infer hierarchy and connectivity of the small molecule and generate the corresponding HELM notation. Multi-HELM can be useful for researchers in the fields of drug discovery, biotechnology, and academic research, wherein the representation of analysis of complex macromolecules is critical.
Thanks, Joanna. I will go ahead and share my screen. So hopefully you are seeing the right thing here. Yes? Okay. So yeah, thanks, Joanna, for the introduction. As Joanna said, my name is Scotty Blechman, and today I'm going to be talking a little bit about Scilligence's multi-HELM tool in RegMol. So Scilligence has always kind of been an industry leader in the area of HELM support, and I think we're very proud to kind of present this tool that helps companies that may still be using old legacy systems adopt HELM format. So I do want to start with just a brief introduction to HELM. I'm sure many of you are already familiar with HELM, but just to kind of get everyone on the same page, I want to briefly go over exactly what HELM notation is and why it's needed. I'm going to talk a little bit about the multi-HELM tool developed by Scilligence. So this is not a brand new tool. but it is something that we've done a lot of active development on recently and are looking to continue doing active development on. And I'll talk a little bit about why such a tool is needed. I'm also going to go over a case study of a client we worked with who had used Multi-Helm as a kind of major player in their data migration. And then lastly, I want to talk about kind of future directions for where we want to go with the tool. So starting off with a brief introduction to HELM, like I said, as Alan mentioned, HELM is the hierarchical editing language for macromolecules. So what do we mean by hierarchical? So the hierarchy starts at the atom bond level. In this example, I've shown glutamic acid or glutamate. So the atom bond structure is going to tell you what atoms are connected to one another, what bonds connect them. But what it doesn't tell you is how can this structure be used in an actual biopolymer. So the atom bond level is only giving you the information about the monomer structure. So if we go up to the monomer level, this is a screenshot from Scilligence's monomer manager inside Regmol. And what this does is it lets you define how exactly that atom bond structure is going to be used in an actual polymer. For example, I have glutamic acid defined here, and I've defined different R groups or leaving groups where the other monomers in the polymer are going to connect to this monomer, as well as some other miscellaneous metadata such as natural analog and polymer type. What's missing here, though, is how does this monomer now fit into the whole sequence? What's the whole sequence, not just this one monomer? And so then we move up to the simple polymer level. So here I have an example that is just a short linear peptide. On top is the atom bond structure. And then just below that is the pictorial representation of the HELM, followed by the HELM string and the sequence. And so in a case like this, it may appear that HELM is just adding this unnecessary extra text to your sequence because it's something you can kind of interpret relatively straightforward when you are given the sequence. But where HELM really starts to shine and where we really start to see the need for HELM is when we get to more complex polymers. So this is one example. I've taken the peptide from the previous slide and just cyclized it. And this is not even a particularly complex case. I've added one piece of complication, just cyclizing the peptide. And yet, if I just try to call this the cyclic version of that sequence, I've already introduced multiple questions. Which monomers are connected in the cyclization? Are the monomers connected from N terminus to C terminus, or are they connected by their side chains? There's a lot of information that you can't interpret in just plain text. And so this is where HELM really shines by explicitly defining for you in the HELM string which connection points of which monomers are connected to one another in a machine readable format that can be processed by automation and AI tools, as well as by Scilligence Regmol. So here's just one more example of another more complex polymer. In this case, I've connected my cyclic peptide to a single-stranded oligonucleotide using a PEG-2 linker, which is called a chem in HELM. And again, you can see the HELM string is explicitly defining for you what your connection points are, which monomers are connected to one another, information that would be very difficult to just describe verbally. So that's HELM. What I want to talk about next is Multi-Helm, which is, like Joanna mentioned, a tool that Scilligence has developed to help companies adopt HELM notation as well as for day-to-day use. So this is what the Multi-Helm UI looks like. You can enter your atom bond structure, and just with the click of a button, it is converted to our pictorial HELM representation. You can also copy the HELM string out of there, or even open up the HELM web editor, which we developed. And we are also able to do auto pairing here. So you can see the small molecule representation does not have any pairing information. And when I've converted to HELM notation, I've been able to just use the Scilligence auto pairing tool to auto pair even with some non-conventional nucleotides like inosine. So what is multi-HELM exactly? Multi-HELM is a mol-file to HELM conversion tool. It's made to make it easier to take just an atom bond structure and identify exactly what the HELM string should look like accordingly. Reverse conversion, of course, is also available, HELM to mol. That is very much the easier part. You have your HELM string, you look at your monomer definitions, and you expand them out. But the real challenge of conversion between HELM and mol file is in this direction, mol to HELM. So our multi-HELM tool uses algorithms to cut the molecule into fragments at certain points and identify them based on the defined monomer library in Scilligence's monomer manager. So this is really the biggest challenge that multi-HELM aims to address, is determining in an automated way which kind of bonds of the molecule to cut to get your individual monomers and figure out your connections. Multi-HELM currently supports both oligonucleotide and peptide structures. Originally, it was just for peptides, and we've since expanded support to oligonucleotides. In the future, we're also hoping to support peptide oligoconjugates as well as chem monomers, which are kind of chemical modifiers that don't fit into either category. And we also have added support in recent years for duplex oligonucleotides. As for inputting structures into multi-HELM, we like to keep it flexible. You can free draw an atom bond structure in the multi-HELM box, as I showed in the UI on the previous slide. You can also upload input structures, mol files, smiles, INCHI, ChemDraw files, anything that is supported by our JS Draw Sketcher is supported by multi-HELM. And possibly even more importantly, we have RESTful API calls available to multi-HELM. We are seeing more and more in recent years that people are preferring to interact with these types of tools via API as opposed to just through the UI. And so we've made sure that everything that works through the UI for multi-HELM is also functional through our RESTful API calls. So why did we develop multi-HELM? I touched a little bit on this, but HELM is a relatively young and evolving technology, but it's increasingly becoming a standard. So HELM, I believe, was started in 2008, and it is now kind of the standard notation for oligonucleotides and peptides. It's been used in a lot of different publications, and it's used across a lot of organizations. And it has the potential to replace dozens of legacy formats that lack interoperability. So lots of companies prior to HELM notation had their own legacy formats that just did not translate between one another, that were only understandable to people within the organization, and it just failed to kind of foster collaboration. So HELM is kind of looking to address that challenge, and we want to help people adopt HELM to address that challenge. A lot of older software tools don't have HELM support, and so a lot of clients may have been working with mol files or other notations prior to moving to Scilligence regmol. And so this is a way of helping people get on board with regmol and HELM. Legacy data sets are also often in mol file format, again, because older software tools often don't have HELM support. And we also have the case where clients may work with mol file on a day-to-day basis, but need to generate HELM for registration in regmol or for their publications. So people may work with mol file day-to-day because they need to see the atom bond structure, they need to look at individual sections of the molecule, but when they are going to register or when they're writing up a publication, they don't want to publish the entire structure expanded because it's more information than people need. Additionally, multi-HELM has the potential to allow for easier structure sharing within organizations. If you imagine a very large biopolymer, you don't need to expand the entire mol file and send that to your colleague. You can condense it into HELM format using our multi-HELM tool and just send the HELM string to your colleague, who can then very easily do that reverse conversion. So I also want to discuss a case study. This was a project that I personally did a lot of work on, helping one of our clients kind of get on board with HELM by helping them with a legacy data migration. So in this project, the client had oligonucleotide and peptide legacy data. They had thousands of oligonucleotides and peptides, all in mol file format with no sequence information. These oligonucleotides and peptides were made up of both natural and non-natural monomers, and there was no base pairing information present on the duplex oligonucleotides. So this kind of presented a unique challenge that Multi-HELM was equipped to handle. The client wanted to adopt RegMol because of our strong HELM support. The previous registration system they were using did not have HELM support, and registering these structures in HELM format would allow for them to be more readable through the pictorial representation, be more searchable by searching by what monomers are present in the structure instead of having to do small molecule substructure searching, as well as organizing their data, again, based on what monomers are present and how they're connected, instead of looking at every atom and bond. Additionally, their duplex oligonucleotides needed to be auto-paired upon conversion to HELM. And that was also something that we were able to help with. So in this particular case, we helped the client first import their monomer library into RegMol. This was a relatively straightforward task because our monomer manager supports upload of SD files or JSON. And so we were able to just get the monomer definitions from the client and upload those into our monomer manager. After that, we wrote a script to process their mol files and send them to Multi-HELM via the RESTful API call that I mentioned. This was done in an automated way such that we could just start the script and leave it running until it was done processing the data. And what we found was that for over 95% of the records in the client's legacy data, we had seamless conversion to HELM notation. So all of the parts of each structure were identified as monomers and identified, more importantly, as the correct monomers. As for the remaining records, HELM has support for something called inline smiles, which is essentially a way of allowing you to include sections of the structure that you don't necessarily want to define as a monomer, sections that are kind of meant to be free drawn because they're only ever going to be used once. And so the way Multi-HELM handles fragments that it can't identify as monomers is it converts them to inline smiles. So because we were able to get that conversion from Multi-HELM with the inline smiles, we were actually able to analyze this small subset of less than 5% of the records to determine for each record which cases were kind of meant to have inline smiles, were meant to have free-drawn portions of the structure, as well as, even more importantly, which cases were kind of structures that we saw repeatedly used across several of these records that were kind of missing from the monomer library. So having this kind of small subset that had partial conversion allowed us to look at that small subset, which is much more manageable than looking at the entire data set of thousands of structures, and kind of figure out which cases were kind of gaps in Multi-HELM functionality, which cases were missing monomers in the client's library, and which cases were intentionally inline smiles. So as for our future plans for Multi-HELM development, as my colleague Alan mentioned, conjugates are a huge up and coming kind of player in recent years in the pharmaceutical world. And so definitely our top priority is adding support for conjugates. In particular, peptide oligo conjugates will be relatively straightforward for us to add support for. We also want to look into adding support for peptide chem conjugates, oligochem conjugates, as well as peptide oligochem conjugates. So cases like the one I showed earlier, where you have a chem linker between your peptide and your oligonucleotide. Additionally, we want to support kind of more complex and rare cases. So things like having cyclization between 3 different monomers via a linker, where your linker is connected to each of those 3 monomers, kind of holding the whole structure together. We're looking to support hairpin oligonucleotides, where the kind of middle of the sequence wraps around and forms a hairpin. As well as unconventional oligonucleotide backbones, for example, glycol nucleic acid, which is kind of drastically different from a ribose backbone and thus needs to be identified differently in the algorithm. So that is all I have for today. Thank you for listening, and yeah, I will take any questions, or I guess I can look at the Q&A. So Scott Starry asks, does V3000 convert to HELM with the multi-HELM tool? Not sure how this would be represented in HELM. So yes, so in Scilligence's monomer manager, you can define each monomer in mol file format. And so that includes support for V3000 because Scilligence Regmol supports V3000 mol file. So the V3000 mol file itself is not in the HELM string, but it's in the definition of the monomer. Frank asks, any interest in including HELM translators in Data Warrior? That is not something that we are currently looking into, but it's definitely a future direction we could go with the tool. Any other questions? If not, I will hand it back to Joanna to introduce our next speaker.
Thank you so much, Scotty. So our next presentation for this afternoon's webinar is Lipid Nanoparticles in RELN. And this presentation will be by Kevin Peuler. Kevin is a senior application scientist who has been with Scilligence for close to 3 years. He has a background in biomedical engineering with nanoparticle research and pharmaceutical experience. For this webinar today, Kevin will be highlighting an exciting research direction with a number of advantages over traditional therapeutics. So with this, we hope you can delve into the experimental workflow and parameters captured in our ELN. Kevin, you can now share.
Thank you. To confirm, you can see my screen, correct?
Correct.
Good. All right. So yes, I'm Kevin Peuler, and I will talk to you about the lipid nanoparticles in ELN. So I guess the first question is, why lipid nanoparticles, and what makes them an intriguing drug delivery system? So I'll go into the advantages, which I'll start with the targeting capabilities. So surface motifs allow for targeting specific cells. And with this targeting capability, you can reduce off-targeting effects. So there'll be less side effects to your drug and less drug would be needed. Lipid nanoparticles have a great half-life. So oftentimes the nanoparticles will have a PEG surface modification, which will allow for immune cell evasion. They're able to carry multiple types of payload, and this does include mRNA, which often undergoes degradation in the body, but the nanoparticles can protect this cargo from degradation, allowing for a therapeutic that is usually very hard to use. They have improved cell uptake, and with that, you can control when the drug is released. So you can allow the nanoparticle to be uptaken by the cell and then release the cargo once it's in the cell. And finally, there is FDA approval. So the FDA is already approving such drug delivery systems, and you can see that with the COVID-19 vaccines by Moderna and BioNTech, Pfizer. So what would we need to capture for our ELN system to ensure that when you're developing these nanoparticles, you would have everything that you would need that you would normally capture in a notebook? So there are different things to consider that you'd like to have captured. You can have your components of the ELN or components of the LNP, the preparation methods, and additional design factors that you might want to take into consideration. So in general, lipid nanoparticles are composed of a lipid mix, and most commonly now is nucleic acid payload. So the lipid mix can include an ionizable lipid, phospholipid, helper lipid, stabilizer lipid. Several include cholesterol or a modified cholesterol, but doesn't necessarily have to include all those. Lipid nanoparticles are very modular, so they can be mixed, matched, modified. So as you can imagine, you can have the same components, but adjust how the formulation is made, and it'll result in a very different nanoparticle that may have different characteristics that you're looking for. So you could adjust the size, the surface potential, any motifs on the surface of the nanoparticle, all to try to meet your need and provide a good drug delivery system. So looking at how these nanoparticles are prepared in the lab to help inform the development of ELN experiments. In general, you prepare a lipid mixture in ethanol or a solvent, and then at the same time prepare the nucleic acid in an aqueous buffer, usually at low %, and then rapidly mix the two solutions together, most commonly with a microfluidic device. And if a microfluidic device is used, then the flow rate ratio between the lipid mixture and the aqueous buffer is really important to keep track of. And then after you've made your nanoparticles, you probably want to remove some particles that are not the greatest size. So there's a number of different methods, such as extrusion, for improving your PDI. So additional factors for your LNP design can include the lipid oligonucleotide weight ratio, or mass percent, because this influences the encapsulation efficiency of your lipid nanoparticle, as well as the nitrogen phosphate molar ratio. So keeping the charge balance between your amine on the ionizable lipid and the phosphate group on your oligonucleotide backbone. And the LNPs commonly have a ratio of around 6, but obviously, if you want to adjust that, that's something that you'd, of course, keep track of in your electronic laboratory notebook. So what is Scilligence's approach to the LNP workflow? So being able to design empty and loaded lipid nanoparticles. We have 2 different approaches for the calculations. So, and they vary based on the values that the user would have to enter. Being able to handle 1 or 2 ionizable lipids in the nanoparticle design, pull component information directly from your registration system. So just like with the ADC, you can register your components, so all of your lipids and your RNA using HELM, and being able to pull that into ELN to design your lipid nanoparticle. And then being able to register your lipid nanoparticle directly from ELN. And they'll be available in 7.3. So to give you a little taste of what the form looks like, I'll walk you through the LNP mole % form. So I won't go into the other form today, but so in general, you'll open the form. And then from here, you'll create your component design. So this includes your API. And then you can design your lipid mix. So this could be 4 lipids, like this example, with only 1 ionizable lipid, or it could be 2 ionizable lipids with 8 lipids overall. It's very flexible, so you can design it however you like. Then you'll enter your batch information, and they'll pull in the molecular weights and purity directly from our registration system. And then specify your N/P ratio that you'd like to have in this design, as well as the flow rate ratio. And then specify your mass and volume of your API, followed by the mole % of your lipids. Of course, you can adjust the ordering of those, but you'll end up with your mass, volume, stock concentration, mass %, everything calculated for you so that you're all set to go with your experiment. This example is a loaded nanoparticle. If you're making a non-loaded one, then you would need to know the volume of your aqueous buffer, of which we can capture here instead of directly in this line with the API. So the next step in the process is creating a registration form. So there is a major challenge for this form with how complex the formulation can be for lipid nanoparticles. So the uniqueness check we've worked on is able to handle a wide variety of components with varying mole %, and you can vary the mass %. And of course, if they have different mole %, then it can be a different nanoparticle. Same as mass % with your design. And then we're also capturing a number of different fields that you would like to have with your batch, such as your size, PDI, zeta potential, encapsulation efficiency, as well as what the buffer is composed of, your solvent, and so on and so forth can all be captured right there in your form. So you might be wondering when these will be available. Well, the ELN capabilities will be available in 7.2, which is currently slated for July of this year. And the regmol capabilities are slated for 7.3, which will be around November of this year. And with that, I can answer any questions, and thank you.
Thank you very much, Kevin. Just want to give a gentle reminder to our attendees that if you have any questions, to please drop them in the Q&A box below. If not, please reach out to us at info@scilligence.com if there are any questions that you may have for any of the presentations. And with that, I'd like to introduce the next speaker and presentation. So the next presentation we have for today will be focused on AI-powered cell counting and labeling inside of the Scilligence ELN. And this presentation will be by Jason Ham. Jason is a front-end software engineer who joined Scilligence just under a year ago. Prior to joining Scilligence, he obtained a degree in human biology from Cornell University and managed a cognitive neuroscience lab with a research focus of abstract concepts in natural language for 3 years. His current interests include applying machine learning techniques to scientific discovery. So as a brief summary for the presentation, to assist scientific discovery in the new era of AI and machine learning, Scilligence has begun developing a new tool for biologists to automatically count and label cells within the Scilligence ELN by simply uploading an image of a cell inside of a cell slide. So in this presentation, Jason will be describing using an advanced object recognition technique. He'll be using advanced object recognition techniques. Cells are detected and identified in a matter of seconds. So with that, Jason, you can share.
Thank you, Joanna. Let me just share my screen. Okay. You got your other. All righty. So I'm going to be kind of shifting gears for the talk, away from chemistry for a second to biology. As Joanna said, my name's Jason. I'm excited to talk about some of the new AI stuff Scilligence is working on. I've been working with one of my colleagues, Ray, to work on this really cool tool for ELN. So AI is everywhere now. You hear it every week, every day. ChatGPT, machine learning, all that fun stuff. And the lab's no different. AI is coming to the lab. It's going to be the future. And Scilligence is joining that craze and moving in with it. So the angle we took for this project is to kind of automate away the things that biologists don't really want to do and need to get done and be more accurate with it, specifically when it comes to cells. With our tool, you'll be able to prep a sample as you normally would, upload an image to our ELN. Our model will analyze the sample image and then it'll return in a few seconds the cell counts, cell labeled, with the overlay of the labeled cells, and then you can finalize the data. So save a bunch of time, increase your accuracy. And so as I kind of went over these steps right here, but then also, as I just mentioned, it'll minimize human errors in subjectivity when you're counting the cells. You don't have to have different lab techs or a scientist counting the cells in different numbers. It'll just be the same number with the model. You'll also free your time, as I just mentioned, so you can actually work on the actual science you want to do, not just sit at your bench and count cells all day. And another cool thing that's a little different that a lot of the other cell counters that are out there don't do is it'll also phenotypically catch cells that you may not have otherwise been looking for. And I'll get into that in a little bit. So I'm going to go through 2 use cases for this. The first one's going to be a complete blood count, which is done every day and around the world, thousands of times a day. It's just a general test to get to assess a sample to see how the specimen's doing. You can use blood count to get an idea of any sort of anemias that might be happening, any sort of blood infections, cancers. And so our model can do this, and it can assist with this. So our model can get a quick, a few seconds count of red blood cells, white blood cells, and platelets to help you assess sample. Over here on the right side is an output from the model. As you see, it right there, it labeled all the cells it saw, gave a confidence interval or a proportion that the model was certain that was correctly labeled. And I'm going to go through in a second here an actual video of the workflow. So I'm just going to play this. So right here, we're in our Scilligence ELN. And we have our template here. We just named example cell model for this. And we're just going to add an image. Upload it. The screen capture software I used didn't capture this other window for some reason, but I just selected a file for my downloads. There's our cell image we're going to use. Over here on the right, in the right column, or left column, sorry, you can label the sample and also look to the counts in the model in a second. So right here is going to give us a generic name for your own housekeeping. You could do whatever you want to, whatever kind of system you want to name this as. We're going to get to the fun part here, where we're going to now select our model we want to use. Oh, we need to select the actual item. Yep. Now we're going to do the model. Again, for some reason, the screen recording software I used didn't have this drop-down menu, but there is one there. I just selected the blood cell model. And now it's going to have the model analyze the image. For this demo, this is still a dev environment, so it's going to take about 10, 15 seconds. In production, this will be under 5 seconds, 3 seconds. Yep, so there it is. In a few seconds, we have the labeled image. We also have the cell counts. As I mentioned before, each of the boxes has a proportion, which is how confident the model was that the cell was properly labeled. Yep, and we're going to go over this 1 second. Yeah, so kind of going over this. And what's kind of cool too, now we're going to, is you can now filter it. So say you wanted to only view, cells which were over a certain threshold. I think I used 0.8 here. Yep, 0.8. And you wanted to do only red blood cells. So what this is going to do is it's only going to show cells for red blood cells and have a confidence of over 80%. So it helps you get a different look at different data, visualize in different ways. We're going to do it again. So we're going to do 0 here, I think. Yep, 0. So this is just going to show all the white blood cells. There's the white blood cell. And I think we're going to do another one for the platelets. Yeah, right there's all the little platelets. And then we're just going to show them all again. So just again, the complete blood test or blood count is run multiple times every week. It's really used throughout every day. And using a tool like this would speed up that process dramatically, save a bunch of time, increase your accuracy. Yeah. So now we're going to kind of switch gears to another use case. Oh, why is it? Oh, stop. Okay. Which another to do more of a phenotypic diagnosis. So instead of just getting an assessment of our sample, we would be using the phenotypes or morphology of the cells to catch pathologies. Yeah. So in this example, we're looking through acute lymphoblastic leukemia. Excuse me. With these kind of things, time is one of the most important things in a medical setting. Time with cancer is probably one of the most important things you can have, is catching things early on. In a research environment, this can save you a bunch of money and resources if you have a sample that suddenly has cancer that you didn't intend to have cancer. Save you time and resources that way. It's also very labor intensive and error prone process. And so this kind of model would expedite that process, streamline it. What's also kind of cool is, right, you could use this in different ways. So you could even just not even use this to intentionally seek out leukemia, let's say. You could have this kind of as like a watchful second eye, where, if you have someone that's trained on, just blood cells, let's say, but not on leukemia, they might not notice that there's leukemia in this data set. So then the model could be used as kind of an alert system where it's like, oh, hey, you should go take a second look at this. There might be some abnormality. And as we'll get into a little bit, this is just an example. These past 2 examples, we can adapt this to all types of morphologies and phenotypic uses. These are just 2. What's also kind of cool with this approach is the examples I've gone through have been about public domain data, which we've used to train our models, but we could also incorporate this with private data or specific patient data to create a very specified data set. We could train a model with particular patient clusters of lymphoblastic leukemia, make our model particular in a certain way that you want to stratify and find specific differences, and have clinically relevant output or results that we wanted to target. So again, this is kind of just a use case for it. There's endless uses for this in different ways. So again, I'm just going to show a quick recording of the lymphoblastic leukemia. It's a little shorter. It's not going to go into all the filtering. We just saw that in the last one, but it's the same kind of process. You just upload an image, and you choose from the models. You choose, instead of blood cells, we're just going to do the acute lymphoblastic leukemia data set. Okay. And again, this will be faster in production. Yeah, so right here in this data set, it's kind of binary. This will be different in use case when it's finalized, but in this case, the model either only tags them as all acute lymphoblastic leukemia or normal. This will be incorporated into the general blood data set, so it would be more specific with the red blood cells in the background. In this case, it was just tagging leukemia. Yep. And this kind of say this is the beginning, right? As I mentioned a few times, this is just a few use cases of it. We're going to be improving user control immediately with it. So you'll be able to, as you probably saw, there were some cells that were missed or maybe maybe the bounding box wasn't perfectly around the cells, or maybe it labeled something wrong. And so the user will be able to go in afterwards with our filtering tool and add boxes, remove boxes, resize them. And so that way you can use this tool kind of as like a way of getting a quick rough draft or of the cell slide, and then you can have your scientist just finalize it. So it speeds up the process that way. We're also going to be expanding the models. We already have a few types of anemia in the pipeline. We have sickle cell anemia, an iron-based deficiency anemia, I think a malaria one as well. So a bunch of different types of phenotypic based diagnoses. We're going to be expanding this to. And we can adapt this to client needs and requests. We're happy to work with you and figure out how we can apply this to your use cases. Just again, for the last time, this is just two examples of, as just a cell count and leukemia, but you can imagine there's a bunch of different types of ways we can integrate this or use this throughout our applications. So yeah, that's all my talk. It's kind of brief. I'll be excited to answer any questions anyone might have. And if not, I'll be passing this over to my colleague, Senna. Thank you.
Thank you, Jason. So I'm happy to introduce the final presentation on our agenda today, and that's going to be speech dictation in Scilligence ELN. This presentation is by Senna Shikoniuk, and Senna is a scientific software engineer who has been with Scilligence for just about a year now. Senna studied biology and computer science at Northeastern University and has experience in protein analytics. Users will have in this presentation, Senna will describe how users will have the option to dictate speech as input for text fields in our ELN application. The spoken input can include higher level biochemical languages, as this implementation has the ability to recognize scientific terms. With a button click, audio is captured through the microphone and translated into the text field.
Thank you, Joanna. Let me share my screen now. Okay, here we go. So now I will be introducing how I implemented speech dictation in our ELN application. So developing a hands-free ELN capability. So the question we proposed to start us on this path was, can machine learning-based algorithm translate scientific language in terms of voice to text accurately? We're moving towards a more hands-free ELN for various reasons. The first being that typing in the lab is frequently not practical. It poses the risk of cross-contamination, and certain settings really require a user to keep their hands free. Because of this, some users can't bring their laptops into the lab. So speech dictation is the start to the solution. This will greatly improve process optimization, as the capabilities of the model we use can minimize mistakes. This more hands-free format will allow a user to multitask in the lab and will almost act as a lab assistant, helping the user through their voice. So this is a general outline of the process of speech recognition in ELN. So I'm here to present the initial findings of speech to text in ELN. The first step was implementing a model that could actually answer our initial question and handle those higher level scientific terms. The next step was integrating our model into our ELN application. And then the next steps will actually be ELN application navigation through speech recognition, integration into other apps, mobile app development, testing, product launch, and so forth. So this is the actual model we're using. So the automatic speech recognition system we implemented is actually OpenAI's Whisper, If you're kind of up to date, I'm sure you've been hearing a lot about OpenAI with ChatGPT, DALL-E, and so forth. This model actually has a word error rate that's comparable to Google, AWS, Microsoft. Those are all actually around like 4.9 to 5, while Whisper actually has a 4.2% word error rate. But the main reason we implemented it was for its ability to handle higher level scientific terms and IUPAC names, making it perfect for lab settings, procedure boxes, and more. It also has the ability to use proper punctuation when you dictate, and it can ignore most of the background noises that can frequently be happening around you in a lab. So test deployment in our ELN application. So the process of selecting a model was actually one of the biggest challenges. We went through various other models, including Mozilla's Deep Speech and Facebook's Wave to Vec, but even with the additional fine tuning and training with all sorts of higher level scientific languages, the models were still not reaching a usable word error rate regarding all of these scientific languages. So then once we arrived at OpenAI's Whisper, the next challenge was capturing optimal audio in the browser. Speech recognition models typically require audio files in certain formats and with various parameters passed to them as well. So we had to actually capture the format and format it properly for Whisper to be able to process. And now here's an actual visual of the user interface of speech dictation. I have a screenshot here of an experiment notebook open here in ELN. Just for this demo, I'm using a button click to capture and record audio for visualization purposes, but in the future, this can change. And now I will show a quick video of this exact model in play. I'm hoping the sound will be able to share as well, but I'll give it a shot. As mentioned earlier, 3-bromo-2-aminopyrazine was chosen as a versatile intermediate. The established methods for the synthesis of bromopyrazine involve the treatment of aminopyrazine with molecular bromine and acetic acid, bromine and pyridine, etc. However, bromination with NBS proved to be a more efficient protocol among all the methods available. Okay, so here you can see the actual dictation that was just transcribed from the audio I captured. Hopefully you guys were able to hear that as well, but you kind of see a bit of an IUPAC name in play here, as well as lots of other higher level scientific terms. Okay. Oh, shoot, sorry. And now at future development. So our next steps in play will be actually the ability to navigate the ELN application through speech recognition. So this would be actually using voice commands to get around the ELN application. So moving from tab to tab, moving to different parts, selecting things and so forth. We also plan to integrate into other applications such as inventory. So this could be very helpful for sample management, actually using data inputs and more. And then mobile app development as well, so that you can be working in the lab hands-free from a mobile device. You don't always have to be using your laptop. And that is all for me today. Thank you. And I will check to see if there are any questions.
Thank you very much, Senna, for the talk. I'm not seeing any questions at the moment. As Joanna mentioned earlier, if you have any questions, please don't hesitate to type them in the Q&A feature of our webinar. All right, it seems like there might not be any questions at this moment. So I wanted to thank the participants for joining our webinar today. I hope that you were able to, thank you, Joanna. Thank you. I hope you're able to get an idea of what we're working on here at Scilligence. I think it's very exciting. We're future-proofing our product, and we have a lot of young developers that are really taking advantage of the emergent technologies that are coming up rather quickly in our industry. So, as you saw, Jason and Senna's use of AI to enhance potential productivity for both biology and also for speech to text features in the future, I think are quite exciting and unique ventures into something new that we could utilize on our product. Of course, Kevin's talk on lipid nanoparticles really points out a key missing need in the software industry to capture that type of modality with the lipid nanoparticles. Scotty's excellent talk about mold to HELM capabilities, and in particular the case studies, how we help our clients address the migration of data that is unstructured or worse, is not HELM optimized, really goes to show how that HELM technology can go a long way to help the industry, to help our clients, and to boost innovation. And of course, we always start working on our existing products. We don't rest on our laurels, as I showed you earlier with the new features that we have on our Conjugate builder. We have plans to keep expanding our capabilities and to keep enhancing our existing features. So with that, I thank you again for joining us. Until next time, take care. Thank you.